Hi friends, my name is Nandita. I am from Andhra Pradesh.
Good morning dear students. Today we are going to discuss about body fluids and circulation. CBSE syllabus class 11 biology. Children, now let us discuss about blood groups. Children, already we discussed that blood is a fluid connective tissue and the most crucial component of the circulatory system. In a healthy person, approximately 5 liters of blood circulate throughout their body. Composition of blood is rather interesting. It consists of erythrocytes, leukocytes, platelets suspended in plasma along with the millions of different molecules with its own specific roles and functions as we discussed earlier. Even though components of blood are the same for all humans, there are various blood types. In fact, there are more than 40 blood groups, but all of them are not clinically significant. The discovery of ABO blood group created great excitement, and until then, all blood had been assumed to be the same. Children, now let us discuss about blood group system. Carl Landsteiner, an Australian scientist, discovered the ABO blood group system in the year 1900. In his experiments, he mixed different blood types and noted that the plasma formed certain blood type produced agglutinates and or formed clusters which were caused by the absence of molecules on red blood cells and resulting in antibodies to defeat that molecule. He then made a note of the agglutination and divided the blood types into 4 different groups. For his the discovery of ABO blood group, he has awarded the Nobel Prize. Blood group system is pivotal in blood transfusion. Our immune system recognizes another blood type as foreign and attacks it is introduced in the body, causing a transfusion reaction. Any inappropriate match with the RH and ABO blood types causes the most serious and life-threatening transfusion reactions. Therefore, before blood transfusion, it is suggested to have a blood group check. Children, now let us discuss what are ABO and RH blood groups. During the blood transfusion, the two most important group systems examined and are the ABO system and rhesus system. The ABO blood group system consists of 4 types of blood group A, B, AB and O, and is mainly based on the antigens and antibodies on the red blood cells and in the plasma. Both antigens and antibodies are protein molecules in which antigens are present on the surface of red blood cells and antibodies are present in the plasma, which is involved in defending mechanism. On the other hand, the RH blood group system consists of 50 defined blood groups antigens. In the RH system, the most important antigens are D, C, E and A. The ABO and RH blood systems are discussed in detail below. The basis of ABO grouping is to 2 antigens, antigen A and antigen B. The ABO grouping system is classified into 4 types as we discussed earlier on the presence or absence of antigens on the red blood cell surface and plasma antibodies. Children, let us discuss about blood group A. People with blood group A have A antigens on the surface of their red blood cells and produced antibodies against group B antigens in their plasma. They can receive blood from individuals with blood group A and O and can donate blood to individuals with blood groups A and AB. Now let us discuss about blood group B. Individuals with blood group B have B antigens on the surface of their red blood cells and produced antibodies against group A antigens in their plasma. They can receive blood from the individual with blood group B and O, and they can donate blood to individuals with blood groups B and AB. Let us discuss about blood group AB. People with the blood type AB have both A and B antigens on the surface of their red blood cells but do not produce antibodies against either A or B antigens in their plasma. They can receive blood from individuals with all types of bloods like A, B, AB and O, making them universal recipients. However, they can only donate blood to individuals with blood group AB. Blood group O. Individuals with blood group O have neither A nor B antigens on the surface of their red blood cells but produced antibodies against both A and B antigens in the plasma. They are considered universal donor because their blood can be given to individuals with any blood type like A, B, AB blood group and O. However, people with blood group O can only receive blood from individuals with the blood type of O. Children, now let us look into this chart like blood type and receive blood and donate blood. Children, the individuals, those who are having A positive blood group, can receive blood from A positive, A negative, O positive and O negative, and they can donate their blood to A positive and AB positive people only. And the blood group B positive people, they can receive blood from B positive, B negative, O positive and O negative, and they can donate their blood to only B positive and AB positive people. Blood group AB positive, they can receive blood from all types of blood groups and they can donate their blood only to AB positive people. Blood group O positive. Blood group O positive people can receive blood from O positive and O negative and they can donate blood to O positive, A positive, B positive and AB positive. Blood group A individuals. Group A individuals can receive blood only from A negative and O negative and they can donate their blood to A positive, A negative, B positive, AB positive and AB negative. Blood group O negative. O negative can receive blood from only O negative people and they can donate their blood to all types of blood groups. B negative. B negative individuals can receive blood from B negative and O negative and they can donate their blood to B positive, B negative, AB positive and AB negative. AB negative. AB negative people receive blood from AB negative, A negative, B negative and O negative and they can donate blood to AB positive and AB negative people. People with the O blood groups are called universal donor because they can give blood to anyone and any people with AB blood group are called universal recipients as they can receive blood from any other group. Children, now let us discuss about RH grouping. A part of the antigens discussed in the previous section, another antigen known as RH antigen, similarly to the 1% in rhesus monkey, is also found on the WBC surface in majority of the humans, nearly about 80%. Individuals having RH antigen are called RH positive and those without RH antigen are called RH negative. RH group is mandatory to be matched before transfusion of blood. An RH minus person, if gets exposed to RH positive blood, will form specific antibodies against the RH antigens. RH incompatibility during pregnancy. A specific case of mismatching of RH group or RH incompatibility has been observed between the RH negative blood group of a pregnant mother with RH positive blood of a fetus, means born out of a marriage between RH negative woman and a RH positive man. In such a case, mother becomes sensitive while carrying a positive baby in their womb. The reason is that some of the RBC from the developing fetus enters into the bloodstream of the mother during development. This causes the development of anti-RH antibodies. The RH antigens of the fetus do not get exposed to RH negative blood of the mother in case of the first pregnancy because two blood remains separate by the placenta. However, during delivery of the first child, there occurs a possibility that maternal blood may get exposed to small amount of RH positive blood from the fetus. Hence, preparing antibodies against RH antigens in her blood, those in case of her subsequent pregnancies, RH positive fetus get exposed to the anti-RH antibodies. This will leak into the blood of the fetus RH positive and destroy the fetal RBC. This could be fatal to the fetus or could cause severe anemia and jaundice to the body. This is known as erythroblastosis fetalis. In humans, during transfusion of blood, the blood of a donor has been carefully matched to the blood of a recipient before transfusing so as to avoid the problem of clumping, means destruction of RBC. A person with blood group of O can donate blood to the person with any other blood groups. Hence, already we discussed here, this condition can be avoided by administering anti-RH bodies to the mother immediately after the delivery of the first child. RH factors was discovered by Landsteiner and Weiner in 1940 after immunizing the rabbits with the blood of a monkey. There are about 30 or so known antigens on the surface of red blood cells that gives rise to different groups, example MN, etc. Children, now let us discuss about coagulation of blood or clotting of blood. When an injury is caused, then wound does not continue to bleed for a long time and the blood usually stops flowing after some time. It is the natural property exhibited by blood to check the excessive loss of blood from an injury. Clotting of blood is a complex process that involves various steps of its completion. Let us discuss about natural anticoagulants. Normally, inside an intact blood vessel, the blood does not coagulate because the presence of an inactive anticoagulants, that is heparin and antiprothrombins. Apart from this, procoagulants are also present in the blood but in their inactive forms. Now let us discuss about formation of a clot. An injury or trauma causes stimulation of platelets in the blood to release certain platelet factors, which in turn activates the mechanism of coagulation or clotting. Thromboplastin, a lipoprotein, helps in clot formation. It occurs in the 3 steps. Now let us discuss about thromboplastin. Thromboplastin helps in the formation of an enzyme prothrombinase, which inactivates heparin that converts the inactive plasma protein, that is prothrombin, into its active form thrombin. Thrombin thus act as proteolytic enzyme to convert fibrinogen molecules to form insoluble fibrin monomer. This reaction required thrombokinesis, an enzyme complex which is formed by a series of linked enzymatic reactions which involves a number of various factors present in the plasma in their inactive state. Both the changes mentioned above require Ca2 plus ions for their reaction. This fibrin monomer polymerizes two long sticky fibers. The fibrin threads form a fine network of threads called fibrins, in which dead and damaged formed elements of blood are trapped. This finally leads to the formation of a clot or coagulum, which is in dark reddish brown scum formed over the surface of injury. Certain factors released by tissue also helps in initiating coagulation at the time of injury.